to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 10th episode of the Weekly Bioanalysis. What a historic week it is to be having done 10 of these. My name is Dominic Moreno and I'm the Senior Scientific Advisor for all things related to ligand binding assays, biomarkers, and flow cytometry. I've been with KCAS for about 7 years, but I've been in the bioanalytical space for close to 25 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, also Senior Scientific Advisor at KCAS, but my focus is on all things LCMS related. I've been with KCAS about 8.5 months, but I've also been in the bioanalytical space around 25 years.
Today's podcast, as always, is brought to you by KCAS, Kansas City Analytical Services. KCAS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. John and I are senior scientific advisors at KCAS, as we've mentioned numerous times on this podcast. And either or both of us are available to answer any questions you might have regarding this podcast or any of KCAS services. As I mentioned, this is the 10th episode of our podcast, and we are so happy to have you listening. As a reminder, you can follow us on Twitter at The Weekly Bio and at KCAS Bio. As I've mentioned before, we kind of started this whole thing because of social distancing, and this was a way to get away from our normal communication. Even today, John is in upstate New York. I'm in Kansas City. Our marketing producers are scattered about the Midwest. This podcast gives the chance to connect with all of you. We hope it's a chance for you to get to know us as well. And even more importantly, it's for you to get to know KCAS and the services we provide. So this week, as I've mentioned, it's the 10th episode. So we're going to do something a little bit different. We're going to introduce you to our marketing team, Jeremy Burt and Aaron Page. With having over 2,500 views of our videos and growing, we thought it'd be a great opportunity to introduce you to them, seeing how it's week 10, and they do so much to support John and I on the podcast. So, as always, the podcast will be a review of last week's news and resources, and then a focus on a topic of our choosing before discussing any feedback we might have. We constantly look for topics, and we'd be happy to discuss something that you might want us to cover. Then we will wrap up and give you a little teaser for next week. So again, we're very ecstatic to be here. This is part 1 of a series of 4 talking about bioanalytical drug discovery. And this week, we're going to jump right into it with a focus on the discovery CMC side and animal studies. John?
I'll start you off with the agenda. So, as you mentioned, this is going to be a substantial podcast because there's a lot been happening in terms of news. As Dominic mentioned, this is podcast number 10, where our main topic is where does bioanalytical fit in the drug development process, and this will be part 1 of a 4-part series. So, as we move forward, we'll first of all touch on news and resources. There's a lot been happening both in the COVID and non-COVID arena. And then it's moving on to the main portion, which is a touch on animal studies and their part in drug development. Then we move on to, well, we usually do the feedback section, but actually we really want to use this as a chance to introduce both Jeremy and Aaron to our audience. And then it's a teaser for next week. And now it's time to go to the news. And for our first topic, I absolutely refuse to talk about this topic after this week, but let's get it out of the way first, then we're over and done with it. Hydroxychloroquine has now been taken off the FDA emergency use approval based on obviously a number of studies that have pointed to its lack of effectiveness against COVID-19. Dominic, any comments?
Yeah, well, you say that like it's going to be the last time you talk about it, but I just get a sense this is not going to go away. I'm sure there's still somebody somewhere that'll maybe come up with a tea or something, but I don't know.
You say that there are still a couple of things out there. I just this morning the WHO has shelved its hydroxychloroquine trial again down to lack of effectiveness, probably taking in information from other trials, etc. But But you know what it means is, I read last night that the federal government has a something of a stockpile of hydroxychloroquine. Any sense of how many doses we have ready for use that can't be used?
It was like 3 million. I mean, Trump ordered up 3 million of them. And so I think there's tons of them available. Let's hope they go to malarial use or something else. But I'm with you. There's a lot of emerging therapies that are far superior.
Multiply that by 21, 63 million doses available.
Is that what you read? 63 million.
That was on CNN something yet last night. But you know that again, the strange thing is we've got all these studies saying it's ineffective, but there still are plans, according to Endpoints News, and obviously I haven't done anything to verify this, to ship it to Brazil because Brazil's got booming cases. Like, well, what's the point in sending something that doesn't work to a place that is struggling with this virus? So yeah, I would like. I really want to see the back of hydroxychloroquine. I would quite happily see those 63 million doses donated to countries that do need it for anti-malarial or for lupus patients. Cut them a break on their prescription pricing and give them a stockpile of pills. I mean, somehow we've got to get rid of 63 million doses. So yes.
I'm sure it's diluting the market on hydroxychloroquine if there are 63 million. I don't know. That's amazing. And just where is that? Like, what warehouse holds that, right? Like, but let's put this behind us. I think we've exacerbated this topic for so long, and I just know we'll be talking about it again, though.
But on a more optimistic note, there is one small molecule that is possibly showing some value. Again, as part of the recovery trial that was being done in the UK, where there's a lot of different treatment arms, and they actually released data from the dexamethasone arm, which has shown effectiveness in helping people for severe patients who are either already on ventilators or are having severe respiratory problems, and dexamethasone has proven to be an effective, at least within this study, is showing to be an effective therapy for patients in that group. So it's not. Again, it's not a cure. It doesn't do anything for early stage COVID or anything like that. But it's one more drug that can be added to the armory to be used in treatment against the virus.
Yeah, this looks promising, and it certainly makes sense given its structure and mechanism of action. It's a steroid. It's something that can definitely help. It's inexpensive. That's very positive. Everything I've read on it's pretty good. Good news, and something that hopefully we don't jump the gun. And I won't say the previous drug, but let's hope it doesn't go down that path where it. I don't think it's gotten approval, but or has it? I can't tell.
No, I don't think it's got approval yet. But like I say, at least there's one study pointing to the fact that it could be useful. I mean, I think that again, hopefully the mode of use for it isn't wide enough that we're going to be stockpiling millions of doses of it or anything like that. So anyway, it is good news, definitely, because we need good news in this.
Yeah, really interesting. Before we move on to another topic, even in that read on dexamethasone, there is. Did you read at the bottom of this? There's 200 products for COVID treatment and 1,100 trials underway as of Tuesday.
Sure.
And there's more than 130 vaccines. So 200 drugs to treat it and covering 1,100 trials and then 130 vaccines. Wow, that's an amazing amount of horsepower going towards COVID. Not surprising.
I know it'll give us plenty to talk about over the coming months. So we're not going to run out of subjects to talk about, at least on the COVID front. So should we move on to the next item?
Yeah, this was an interesting read. It came out this weekend from NPR, where there's always this mixed bag on how detrimental or how dangerous COVID really is, and well, they compared it to the weapons of mass destruction debacle. Now, I don't know if it goes to that level, but they made a case around the death rate, which and they sort of they wanted to emphasize they're not like Monday morning quarterbacking it. And what I mean by that is they're not saying in June, I told you so. They're quoting studies from early on back in March where it said, they came out and said the death rate is 5% or somewhere between 5 and 10, which is I mean, that is like Spanish flu like and incredibly scary. I mean, I think COVID still is scary. We should still have some degree of caution, but clearly the death rate is nowhere near that. And they even quoted back then that there were other statisticians or other scientists that were saying it was more in the 0.5% to 1%. So thought that was interesting, sort of tidbit around, specifically that it's NPR and that they were kind of comparing it to the weapons of mass destruction debacle of the Iraqi war and everything. So interesting read there, John.
So was that? So basically, saying the infection rate is down at the 0.5%, or is it the fatality rate?
Fatality rate.
Okay. Okay. Sure. Yeah. Yeah. So, because I mean, obviously, we're still in that position where we have to really guard against infection passing on because of what we're seeing around the country and around the globe. Which I know we're going to talk on. We're going to talk on that a little bit later.
Maybe it's a nice segue into the next topic, which is, somehow apparently there's a report saying the protesters are not getting COVID. But I think that's sort of changed even from when we put together the news and notes from 2 days ago. I feel like there's been more impact on protesters coming down with cases, but that's still, I'm sure there'll be more on that, but certainly there was an indication that it wasn't causing spreading because it was outside.
I think outside helps in terms of minimising effects like that, but ultimately it's still early in terms of protests because you're talking a 14-day incubation period. There's still protests going on, so I think it will be a topic that we'll revisit probably as this develops.
Well, I mean, I think, and really I feel like the majority of protesters, at least as the day wears on, I think there's a lot of people protesting during the day, but certainly as the nighttime protesters, or maybe even those that are sort of rioting, those individuals, are they really going to get tested? I mean, I can't imagine they're, you know, unless they're incredibly ill. So I feel like that's a segment of the population, and maybe even myself included. Not that I'm a protester, but there's certain segments that just, despite any symptoms, they're just not going to get tested, right? I mean,
Well, yeah, and particularly if so many people are asymptomatic or they have mild symptoms, it might not drive them to get tested. It probably will be the people who get very sick. It's like, yeah, I've got a problem here, and then go and get themselves tested for that. So I think there is going to be. You're right. I think there's going to be an inclination not to get tested on that kind of thing.
yeah, which segues nicely into the next topic, which is only about 5% has been tested, and really they're not testing people unless they are symptomatic. To your point, um, I think there are cases where people have at least have, some of it might be hearsay where, hey, I came in contact with someone that was exposed, and then the physician says, hey, are you symptomatic? No, well then we're not going to test you. So I don't know how true any of that is, but that's some of what I hear. They're not testing people unless they show symptoms, and I don't know what's right or wrong, but to only have 5% tested, that makes it really hard to understand how many people are asymptomatic and how many people are, potentially had, every time someone had exhibited some sort of symptom, I'm sure they're not getting tested, or I don't know.
Yeah, and tying it to symptoms does seem weird when we've read, and again can't remember the proportion, but there's a lot of asymptomatic cases out there. Where, I think actually that was one article I was reading this week where Florida opened up and there was a group of 16 friends went out to a bar, and they interviewed a couple of them and said, yeah, we weren't really practicing social distancing, but we really wanted to go out, but all 16 of them actually have tested positive for COVID, but all 16 of them so far asymptomatic. So it's like if you, I think that must have been a part of test and trace because of I assume there was someone was infected in the bar, and so it's following up from that. But yeah, to do it. To not do it if there's no symptoms present doesn't seem to make a lot of sense. So I don't know. We'll see.
It's still, and then like what was the test? Is it antibody? Is it viral, right? And then the rate of asymptomatic. I mean, I've seen 20 to 50%. I really to me, it's going to even be somewhat segmented based off of your age too, right? And of course, I think there's some gender issues there too, where men are more susceptible and stuff. So I don't, it might be if you're under say 30, you might be, 85% asymptomatic or 90 or something. And then obviously it would increase with age. And then the other, really, it might be somewhere like you described a scenario where 16 people all tested positive, but perhaps they only got a small bolus of the virus, right? So then it comes into that whole conversation around how many viruses does it take to get to the center of a Tootsie Pop? No, just kidding. But how many viruses might you need, or how big a bolus of a exposure do you get? Is So the other thing that's happening is herd immunity. I don't know if that's where you're going, John, but herd immunity is something that came out recently, and the experts are cautioning that. I think people have an expectation that, oh, look, people are getting sick, and that'll cause this herd immunity. 4 months ago, no one had ever heard of the word herd immunity. Now it's like all over the place, right? But that the reality is, for herd immunity, it's upwards of 70 to 80% of the population, and that could take upwards of another 18 months before we think there's truly like what we would consider herd immunity, where we kind of return to our normal life. So herd immunity is on the same rate as a vaccine. In fact, maybe even the vaccines are ahead of it as we get to some of our next
Herd immunity can also mean a lot of deaths in the meantime, because I mean, you look at countries around the globe and their approaches they took, and Sweden was the one that really went the way of going for herd immunity, and their per capita fatality rate was way up there in terms compared to other countries. So yeah, long term herd immunity will help, but I wouldn't use that as a policy to approach this. It's definitely the other social distancing, face masks, etc., are a better way to go about this.
Absolutely, wash your hands, all of that. The next topic's a real interesting one. I'll sort of tee it up for you. It's this caution fatigue, right? Is that sort of what's happening in terms of like people are getting somewhat stressed and fatigued by all of the changes that have happened over the last 4 months? I certainly can kind of understand that. I think it's something that might affect all of us.
Yeah, I think after a while you sort of there's this sort of mentality where you think, I've taken all these precautions and nothing happened. Well, why should I keep taking those precautions? And obviously, once you let your guard down, if you're out there not taking the precautions that we know to be necessary, then there's a good opportunity to actually get what you were trying to avoid in the first place. Yeah, it's definitely something to watch out for.
Yeah, I mean, I find myself using a mask a little less. I think I'm victim to it. Like sometimes I kind of forget it in the car, or maybe not even take it. Now there are very few places in Kansas where it's required to be worn, so I do keep one in there just in case. But I feel like for me, to avoid that, I just haven't been going out. I've only, we've been open for a good solid 3 weeks. I've probably gone out to eat in a restaurant. I think was the third time. Earlier this week, I had some breakfast with John Buckstaff of all people. But yeah, it's been something where I think my family and myself are really taking a very cautious approach, and not really like haven't been to the mall, which is open here, and I think we're kind of somewhere in the middle on this caution fatigue. But I do think our guards being let down on the mask a little bit.
Yeah, actually, I think probably different state, different policy. I think there's much more focus on keeping the mask on, so it actually prompts us to do it. I think in terms of us as a family, we're not ready for restaurants, etc. My wife's definitely more cautious in approach than me. We've had sort of some social distancing, meeting up with friends and stuff, but you still sort of feel uneasy about, oh, we've done three different things in one day with different people. Is this the right thing to do? So no, we're not there yet. We're still on the cautious end of the spectrum, definitely.
And and except for Menards, where you have to wear it, that's a hardware store out here. You got a home store, you got to wear it. I got to tell you, if you wear a mask, you're kind of maybe in the minority now, as opposed to where we were a month ago here in Kansas. And we're even returning to some sporting things, like my son has returned to some outdoor lacrosse practice. We're being very cautious there in terms of distancing, and then he's started conditioning for football, where they're separated. It's not contact yet, but all of that is, it's been a couple weeks on the lacrosse thing, but as I sit here and talk about it, I'm like, oh my God, am I a bad parent? Like, I don't know. It's all of that comes rushing in, right?
Sure. Yep. Yep. And not talking of sport, English football. I can't bring myself to say soccer. Started off last night, so yeah, there was a couple of games there, but with no audiences following on from the Bundesliga and other leagues that have already been going for a few weeks.
Oh, my favorite. The English Premier League is absolutely my favorite football league to watch. It is that started up yesterday. That's right. Chelsea's my team. Go Chelsea. Let's move on though, John. How about what's happening in China and South Korea? Right, interesting stuff.
Well, again, it's interesting to see just how pernicious this virus is. That you've got countries who have got things relatively under control, but it doesn't take much to really kick up a small spike. So China closed schools in Beijing because of as a number of cases they were seeing there. South Korea's spiking. I can't remember what the specifics were of that, but actually New Zealand had no cases for, I think, almost 40 days, and they had 2 cases recently. It was actually 2 women came in from the UK, and they were granted compassionate leave to go and visit a relation, and as it turned out, both those women developed symptoms, and I'm not sure what New Zealand's doing to contain it. I'm sure that they'll get it under control. I'm sure that both China and South Korea will get their outbursts under control simply because they're so focused on getting it because they've managed it before. But it just shows how even when you think it's all you've got everything in place, it doesn't take much for it to sort of pop up and cause a number of cases again.
Yeah, well, they're saying the spikes we're seeing is still round one, right? That we have not hit a second wave, and I don't know if what's happening overseas is sort of that second wave, but where everything you're feeling now is still, to me, the still part of the initial wave of COVID, right? I don't think.
I think certainly here it is. Again, I know I've mentioned it in the past. The Johns Hopkins site is excellent in terms of showing trend graphs, but countries like South Korea and China and New Zealand all brought it down to 0 new cases. So they got through that first wave. They obviously got to the other side where they could say we've got this under control, and now they're seeing it spike up again. So that to me would be second wave, and yeah, I'm sure because they've got their experience from how they dealt with it before. They'll figure it out. But it's something that every country has to be aware of, and I know we touched on it in terms of there's a number of European countries who are opening up for tourists, etc. They are definitely specifying what countries are allowed in to even visit as tourists, but it only takes one person. It's like that company presentation we had today. The what if case if one person comes in with an infection, how it can really change. How it can really devastate a population.
Yeah, we're not going to solve it, John, but yeah, it is fascinating stuff, and really, it's hard to envision a time when this won't spike until a vaccine comes around. If we're seeing countries that have driven it down to zero and now it spikes back up, I don't know. It gets hard to think about the incubation time and all of it. The asymptomatic, the 14-day incubation time, or putative. That's still a little bit controversial, but still certainly well past 7 days, right? So all of that has, you can see how you think you're in the clear, and then boom, you're hit with.
Yeah, I mean, just to revisit the New Zealand case. I mean, like I said, they were down to zero, but obviously you can't just shut yourself off as a nation. So they've got travellers coming in. I think their approach is going to be doing much more testing as people come into the country, so that they can figure out how someone, if someone has any issues, do they isolate them? And I'm sure there'll be much more conversation on that. But in terms of long term and a vaccine, so let's move on to that. So, sure, absolutely. It makes a change for me to be talking vaccines, but this isn't so much about the vaccines. Obviously, as you mentioned earlier, there's over 100 in development. But what we're seeing is the focus because of the scale of the need of a product once it's approved, the focus isn't just on the development; it's really on the logistics. So for the actual pharma companies, a lot of it is about how do we ensure that we have sufficient supply. So I think it was AstraZeneca already have some capability for producing vaccine, but they've now partnered. They're now partnering with Catalent to help ramp up that production when they're ready to do so. But the other big part of this is actually the way countries are manoeuvring to ensure that they have a supply. So AstraZeneca have basically, I think, have already inked an agreement with the US, but they're also in the process of sorting out an agreement with the EU. Germany has grabbed a minority stake in CureVac, who were previously courted by the US administration, trying to bring them over here so that they could, get first dibs on a CureVac vaccine if it was proven to be successful. Just today, it was mentioned that Johnson and Johnson have also signed an agreement with Europe. So there's a lot of this going on. And also, as part of Europe, there were, I think it was Germany, Italy, the Netherlands, and France had pulled together like a core team of countries to work with pharma companies to ensure they had the supply, with also intent spreading that out to Africa in terms of helping Africa get their supply too. And actually, there was also, just on another topic, was also very interesting that there's the Chinese virus being developed by Sinovac, isn't taking the modified RNA or DNA approach that so many of the higher profile vaccines are taking. It's taking the approach of using an inactivated virus. And at least there was one commentator said they like that approach because it's likely to be cheaper, and if it's likely to be cheaper, then it makes it much more attractive for countries in developing countries who can't afford expensive treatments. So it actually gives a really nice alternative to the other vaccines that are in development. So I've said a lot there, and I'll let you say something.
It's safe too, John. If it's done right, it's safe, right? It's a much safer vaccine than some of the other technologies that are out there. And this is like what polio is: inactive. It's attenuated COVID-19, activated COVID-19. The Sinopharma, they've been vaccinated over something like over 1,120 volunteers already, and they're planning on vaccinating the whole city of Wuhan. So, yeah, I mean, to me, there's no harm in giving it. They haven't really demonstrated its efficacy, but that's one of those. I look at these vaccines and say, would I put it in my arm or would I give it to my child? And barring that they're making it in a safe environment, that's, I'm just going to assume that, yeah, this is something you could deliver. It's really shouldn't, if it's made properly, should not have any adverse effects and could be potentially safe. Yeah, and definitely cheaper than the technologies that we've touched on around what Moderna's doing and others, where those are highly engineered designs that are more, catered towards neutralizing and demonstrating different efficacies than just attenuating it and hoping that those, epitopes are protective in their attenuated state.
Yeah, so just to go back to Sinovac, they said that they were able to safely spur protective antibodies in more than 90% of volunteers involved, but that was just a statement. There was no data to back that up. So again, we'll see that in time.
That's an antibody test, though. So I give you a protein, a foreign protein, 98% of it, you develop an antibody against it. Doesn't make it protective, John. That's like
Understood. I'm just saying their press release said. So we'll see where it goes. But it's just an interesting alternative to the ones we've already been discussed in more depth.
one thought before we move off of COVID is the vaccine supply. It's amazing what's happening. That the Catalent thing that you're talking about, they're going to build a new site in Italy for this. So they're like building sites for COVID specific stuff. And that's, me, John, I love to pump the economy up, that's a great sign. And you know, we'll see how quickly they can get that up and running. But I think this is something where the world won't get caught again not having the capacity to make a vaccine to help save lives, right? I think that's what this is unearthing.
Sure, and one other comment, and we really have to move on from the news pretty soon. But as we're talking about governments all trying to get their hands or guarantee a supply for the future, the World Health Organization is starting to draw up guidelines to actually say who should be priorities if once we have a vaccine to roll out to people. So they're talking frontline workers, vulnerable people, those in higher transmission settings are some of the people that they're identifying as they should be the first to get doses. So I only saw that this morning and thought, right, I'm at least going to throw that out when I get the chance. So yeah.
Yeah, I'll wait patiently. I'm none of those. I completely agree with that. I'll wait my turn. But yeah, I think those people that are at the forefront, give it to them first. Yeah, I'll stay in my basement and hide away till I can get a vaccine. So I'm John. Maybe I'll just quickly touch on the three non-COVID items that we talked about. One is the trial disruption easing, but there's still some fallout. And what that means is we're starting to open up clinical trials, and that's an interesting thing that's occurring, but there's still some hesitation around the impact that it's having on drug development. The other is a nice story on CAR T cell manufacturing. That's an exciting thing. A new facility had opened up. I believe it was Gilead had opened one up in Amsterdam. So that's exciting. And their Yescarta drug therapy.
So I have a question about that. How hard do you scour for CAR T topics every week?
Oh, it's such a passion of mine, right? Like that's, the first like 10 years of my career were all in autologous T cell therapies, and now they call them CAR T cell. So no, I don't scurry, but I keep an eye out for it. Yeah, I don't. It's interesting. You'd be like, I understand. Like, I don't get any. I don't have a specific alerts around CAR T, but when I see them, I'm definitely reading about them. And then the manufacturing challenges are so enormous, John. Right? So these are like you need class 10,000 clean rooms. You need closed systems. You're taking someone's blood and you're manufacturing it in a very aseptic way, and it's there for like upwards of 30 days and stuff. So there's a tremendous amount of QCing all along the way. You got to sample it all along the way. And again, having spent several years doing it, it's something that it just always piques my interest. But I get you. It does seem like I do find them.
I thought you were working on the basis that no podcast is complete without mention of CAR T.
This is a really interesting story. It's the one. This is the first of its kind approval, and we always like first of its kind. The FDA has now cleared for treatment of ADHD a prescription video game. Fascinating stuff. Truly, I think they overdiagnose ADHD, but those that truly, it's an affliction to a number of individuals. This sounds like a wonderful remedy for them. And, maybe it's not so bad that I played all those video games as a kid, John.
It's fascinating, isn't it? I wouldn't never even have dreamt that would be something we'd ever see happening, and yet here we are. It's amazing.
Well, for the sake of time, we're going to skip over any investment stories this week and we'll pick them up next week. Let's move on to our main portion. This week's topic is, where does bioanalytical fit in the drug development process? We're going to do a series on this. It's a fascinating topic for us, John. Let's kick it off with drug development focus is often on discovery and CMC, right? We think that's probably where most people, when they think about drug development, what comes to mind is the discovery and CMC portion of things. But that's part of the equation, and certainly we play a role in those things, not the CMC, but we frequently get pre-GLP material and do research bioanalytically on it. And so, this is the when you think about drug development and the process and bioanalytical, those are such big words that are somewhat vague. So I hope we can start to drill into them and kind of narrow it down a little bit and kind of like, over the next coming weeks, focus in on what that all means. So that's my diatribe to start us off, John. What do you think?
Yeah, I was thinking about this earlier, and yeah, I mean, it. I think it's worth talking about what the drug development process is, the sequence of it, And that's largely how these podcasts are likely to break down. Certainly, we've got discovery, then going into later animal studies, going into clinical. But we also wanted to focus on how bioanalytical fits in that. And as I was looking at stuff this morning, it's interesting that bioanalytical is key up front, but the longer a drug goes on to in development, sort of bioanalytical sort of fades, with the exception of late stage where biomarkers become more important than the actual investigative drug in terms of what they're looking at with people. I don't want to spoil that because obviously we'll talk about that when we actually talk about clinical development. And this is more talking about animals today, but I think that's probably a good way to approach it. So, from a small molecule basis, a lot of larger companies have huge libraries of investigative molecules. So early stages in terms of discovery, even before it's administered, there's a lot of testing to be done even before there's any thought of administering to animals or anything like that. So they're going to test solubility. We can do a lot of in vitro work, looking at metabolism, stability, receptor affinity, and what they're trying to do is hone down as to which of those multiple drugs might be good candidates to go after a specific target. And then they would go to preliminary discovery. And chime in anytime you want, Tom.
No, I was going to stop you for just a moment because the large molecule space, the biologic space, slightly different, right? We got libraries and libraries of monoclonal antibodies. We got all these proteins out there. Our discovery is much different. It's more like it's more around, hey, we got a target against X. You know, when I say X, it could be some sort of receptor on a tumor cell. It could be some sort of soluble cytokine. But that the dilemma becomes slightly different. It's not everything you just talked about there. Those are we can readily make a humanized monoclonal antibody. We can readily have an engineer an epitope against any given site within any sort of protein that we want. The reality is trying to hone in on which one might be effective. And then our processes are a little different around how we approach discovery, right? So you're looking at thousands of candidates and screening. We're more like, we got these 4 that are against this epitope. What do we want to do? So it's a little bit of a different, I think, dichotomy comes to mind when we talk about discovery between small molecule and large molecule, and of course, even on the CMC side of things.
Oh, absolutely, and that's partly what I was sort of wrestling with as I was pulling this together earlier. You know, because I just wanted to have some cheat sheets to chime off. But I mean, so yeah, so basically, if you've got you may have a group of similarly structured compounds going back to small molecule, and then you may have preliminary studies in animals, which would just be pharmacokinetic in nature to see what the how it's doing in terms of residence time within the body, so half-life, etc., and really trying to whittle those candidates down to the ones that go into fully regulated animal studies. And even within discovery, there's going to be various needs of rigor. For very early studies, if you're just comparing different candidates within the class, you just need a quick and dirty assay. So for discovery support, it's going to be a very generic LCMS approach where your LC conditions are pretty much going to be set unless it's a very unusual molecule. And then the mass spec conditions, you'll gear it to the molecules, but it's with a minimum of tuning almost. You won't have really challenging sensitivities not needed. Just trying to get a quick handle on these drugs to really push them forward. And ultimately, the purpose of animal work is to prove that the drug could be effective and safe in humans. So I'm going to touch on beyond the discovery in a minute, but ultimately the discovery piece is just figuring out the drug is doing what it. Well, sorry, not really doing what it's supposed to be doing. Discovery is really just determining what the drug, what its pharmacokinetic profile is like, and sort of bioavailability and pieces like that. And then we move to GLP, where it's more rigorous assessment of what that selected drug is doing in the animals.
Yeah, so I mean, on the look, the term quick and dirty is not something that really exists in the LBA space. There's nothing quick about it. There's nothing dirty about it. I know that's a term you guys use, but really when we're in the discovery space, it's really got to be, a fully developed method, and you can maybe stop there. We call it characterized, but then typically people qualify it even at the discovery level, even if it's a candidate to. Now there are exceptions, and most notably is the monoclonal antibody space, where if you have a humanized monoclonal antibody, you can do, what would be, it's called a universal assay, and that would be the equivalent of your quick and dirty. And what that means is, because it's a human immunoglobulin, you can look to capture just against a base human immunoglobulin in a non-human species, right? So all the circulating antibody in a rat, a mouse, a dog, whatever's non-human primate, that is not a human antibody. So therefore you can take advantage of that. So you just capture any human antibody, and then you come back and detect with it. And that's a universal assay, and that is what is typically used at the discovery level for mAbs. Otherwise, you've got to invest in capture antibodies. You got to get some reagents. You don't have to fully validate them. But on the flip side, the other bioanalytical component that's often overlooked in the discovery process, and it's becoming much, much more mainstream over the last, say, 5 to 10 years, is the immunogenicity component of it, John. So we have a second bioanalytical thing to deal with that's quite different from the small molecule. And you've been drinking from a fire hose since you came here around what that means. And that's all the anti-drug antibody production. And that is certainly something that, you want to make sure you're doing some sort of work, although typically it's still some clinical assessments can tell you how safe the drug is. But you do start to look at production of antibody at a much earlier state than people had been, say, again, 5-10 years ago.
Sure. Yeah. Yeah. And when I say quick and dirty discovery, I mean, ultimately the quantitative assay, our approach is we have 3 different levels. So the quick and dirty is limited number of QCs. So we got the sort of fit for purpose basic LCMS assay, but then even in discovery, we have different levels of rigor. So depending on what the question is that's being asked for, it might be a molecule is out of that just very quick pharmacokinetic screen, but they're doing more preliminary, more additional studies, seeing how it behaves over like a 7-day study. But they might want, the customer may want more rigor that doesn't need to be at that fully optimized assay. So we will include QCs. We have wider acceptance criteria than we typically have for a regulated assay, but there is definitely more focus on ensuring that the assay is qualified before we then touch samples. So, like I said, there's these different. We do run different levels of discovery assays, and also, I mean, the other things that are being assessed in discovery are, the clearance is something that's going to be important. We will look at tissues to say where the compound's going. Another important piece is some any preliminary SIP P450, just in case there's likely interactions to be aware of. How's the analyte metabolized, and that's something we should be looking for in the future. And again, we'll talk more about that in the clinical version of this podcast, which will be the next one. So there's a whole wealth of things that discovery gives you before you then move into the GLP talks.
Yeah, and we have a similar approach. I mean, we don't talk in terms of research grade, but that universal assay, as well as any sort of discovery-based assay, you will have QCs, maybe not quite as many as you might in, say, a validated method, and you for sure can widen your ranges a little bit to help with, you just don't need the amount of rigor of having like a pinpoint accurate method for discovery candidates, and certainly it can be used in the right fashion. And in the universal assay space, the tricky, that method, we have a method. We've used it on probably I don't know, upwards of 20 or 30 different compounds. The key there is how are they making it? So, meaning they might make it in cell line X or in some sort of other system, and how it gets glycosylated will impact that. And that's where there's some development with when we take our method for the first time and put a monoclonal antibody on there, or even an ADC for that matter. We typically need a couple development days to ensure that the capture and detection that we use is optimized, or at least slightly, developed, to ensure we're getting, accurate results. And then after you do it once, you can pretty much plug and chug. Say the same customer comes back with a handful of different antibodies, but they're made in the same system. Then we do almost no development. We can just run them, and sometimes we can even run it off of just one curve instead of say four. But for the number of samples we're doing and so forth, we typically like to keep a curve for each monoclonal antibody that we put on there. I hope that made sense what I'm saying, but that's our sort of pain in the discovery space and how we do what we would consider, quick and dirty.
Yeah, I think with the exception of the very, very, very basic assays, I think it's important even in discovery that you have to be able to stand behind the data. So for us as analysts, the important difference for discovery versus GLP is we don't involve QA in review of the data, but we still have to be able to stand behind the results that we're producing. So it's still critical that the methods are strong and robust. That really is, that's still our driver here.
Yeah, and you have, you can fail runs based off of, at least in my world, it's percent CVs between replicates, any sort of standards and QCs that don't recover properly. Maybe you accept the data, but you really put some sort of caveat on there that says, hey, this should be taken with a grain of salt, or we just repeat it, right? But there's nothing we do that isn't at the GLP level. I think of it as it has elements of it that are GLP, for sure.
And it's a BB makes the point. I mean, the instruments that we use for discovery are the same instruments that are used in GLP work. So they're all fully qualified and calibrated and everything to ensure that they perform at the level they should. So moving on to GLP. So really, once you've selected that, at least back to the small molecule world, once you've selected that candidate, then you really want to have formalized toxicokinetic studies. So again, you're still be looking at the pharmacokinetic profile, but we probably start with 2 week studies, maybe 4 week studies.
Similarly, we certainly on the biologics side, the I think the real difference is a protein. Obviously, its ADME is much different. Its absorption, distribution, metabolism, and excretion is going to be very different than a small molecule. Most of it, when it's given intravenously, there's two places it kind of goes. The lungs is one. I don't know if everybody knew that. The other is the kidney and liver and things like that. So it has a slightly different pathway. And so, some of the things like P54s and stuff we don't look at, but when you give a protein, sometimes a fever is a sign of an animal having an adverse event. I think they look for those types of things. And then obviously if the animal succumbs, which sometimes can happen when you're given large doses of a given antibody that might be against something endogenous or something that might be critical to one's immunity, right? So a little bit different there. I think at an earlier stage, there might be more. Again, I talked about the ADA component, and that's the safety component. So there's, really a sign if you're in the discovery stage and or even like a non-GLP study and your PK profile has some wonkiness to it and you believe your method's good, or there's a subset of animals that have some perturbations of the PK profile, that's a sign that there's actually an anti-drug antibody being made. So some people would argue you don't really need an ADA method. All you need to do is have a really, really super sensitive PK method. And if you see perturbations in that, then it's antibody dependent, meaning it's neutralizing or knocking it down. So I think there's slightly different ways of assessing the safety in the discovery space between small and large. Not slight. I think they're quite big.
Sure. Yes. So for us, yeah, basically we're going to be measuring the drug in the studies. Like I say, it's purely again on that sort of PK basis. But the PK assessment is part of wider studies, and these, like I say, the longer term studies are things like genotoxicity, carcinogenicity. That's what I was looking for as the word. We will do reproductive toxicology studies. So, I mean, they're usually smaller studies, either rat or rabbit. And actually, some of these studies may actually be run in parallel with the actual clinical trial, depending on how quickly your development's moving. Um, but so, all of this is really the intent, because where you're doing it in parallel with a clinical trial, you're doing it in a study with volunteers, so it's not a patient population, it's not a long term study that it's going to be running at the same time. But ultimately, everything in animal trials is geared to ensure that what you're doing going into human is safe.
Yeah, and I don't mean to say we don't like that's still done on the large molecule side. I don't mean to imply it's not. It's just I think there's a different approach to how that's all looked at and measured, and maybe even the number of animals and the number of studies might vary between large and small. But it's all stuff that has to be, that box needs to be checked independent of the size of the compound.
Yeah, so for us as analysts, we're focused on the molecule in terms of the looking at the effect on the test vessel, be it whatever animal that's been done by other teams, and this is where looking at bioanalysis, we're a small part of the development process. There's so much going on around us, and like I said at the beginning, in animal work, an awful lot of what is measured is passed through us as analysts. But as there's still a number of groups doing other critical assessments to test that drug. It's not all just based on the level of the drug in the animal's bloodstream or tissue.
Oh yeah, the in life is definitely doing a tremendous amount, John. Right there, they're taking all sorts of tissue types and mass distribution of it, and all of that is typically, some of it we can do, but again, I think the in life will do a, quite a bit of. I think that still falls under bioanalytical testing. They're looking for drug, they're looking for damage, but they're going to do a tremendous amount on the to sort of. Not counter, to complement the PK work that we will do, as well as any sort of potential ADA and biomarker work that we're doing.
Yeah, so I mean, so for example, I mean, there's things tested in GLP like routes of excretion. We can support that. It's just there are other approaches to do it. Again, quantitation in tissues, that's something we can do, but there are other approaches for looking at the drug in tissues. So yeah, we're a lot of complementary groups working together.
Yeah, I mean, by the time we're doing tissue analysis for the drug, usually we're doing some sort of pharmacodynamic alongside it. I think we're at least the standalone PK in tissue is not as common as it is with, hey, we really need someone that's got a good understanding of pharmacodynamic testing in tissue, which we do, and then it's like, oh, by the way, why don't you test for PK and we'll put it all in one facility instead of shipping the sample, trying to split it. Where the other lab may have been able to do some PK work, and I mean the other is like an in life. They're not. They definitely frequently struggle with understanding the regulatory requirements, as well as even just the nature of a functional test like a PDA assay might be. So, John, as we're starting to wind down here on our main portion, one thing that frequently I get asked, frequently comes up, is for my animal studies, what species should I use? And I don't have a magic answer to that. I think it depends on the drug. There's all these depends. What I do know is you got to do two typically. That's, before you go into humans, you got to do two different species. And for things like monoclonal antibodies, it's like pick a rodent and then do a non-human primate. That, do you want to do mice and non-human primates or rats and non-human primates? And then there is some justification for why you might want to go into a dog or a rabbit or things of that nature. Certainly ocular studies tend to be more rabbit things, but for the most part, it's like, yeah, pick a rodent and then go into non-human primates, right?
I think that's it. Well, certainly be my experience, but I'm not an expert who would ever be driving that choice. But it always seems that's usually the structure of what we're seeing in terms of studies is one rodent and then one larger animal. Like you say, ocular is very much rabbit comes into the fore, and then you will see other species like mini pig come in for specific indications.
Yeah. Mini pigs are good for bladder and things like that. Dermal studies, cardio studies, And I know bladder studies are typically, maybe a pig is a good species for that. But I mean, I don't think there's a right or, I don't know if right or wrong is the right way. You just got to do your homework and leverage what others have done and make some decisions.
Exactly. Yeah, I mean, it's always interesting to see what unusual species come across your desk in terms of please work on this. But we once worked on woodchuck plasma as part of a hepatitis drug in just one study in that. So
That's I mean, woodchuck plasma. We've done studies in rodent breast milk. That was an interesting one. And then that was really very interesting in terms of how you harvest that. And then the other one that I find that we're working on now is ferrets. Ferrets are big in the CAR T cell space, John, right? So they actually, for whatever reason, they're being used for AAV testing, the adenoviral vectors. Ferrets make good models, and I don't know the whole biology behind it, but that is the model of choice for before going into humans. And the AAVs are part of what's typically, not all, but a lot of CAR T cells involve some sort of viral vector, and it can be AAV. So that's, we're heavily involved into looking at AAV, and it's actually an anti-drug antibody or an anti-AAV antibody, and they're doing it in ferrets, and we've got some experience there. So I think, John, what we'll do is, we're going to stop right there. We've got our series, and maybe we can let Jeremy chime in now. But I think what would be nice is that seems to be part one of our series, right? Do you agree, John?
yeah, I agree. I mean, I think we talked a bit about animal stuff. Obviously, we would love to talk more in depth with an expert on some of this, but that's a topic for another time. But the next section of this will be talking about clinical trials and the format they take, and we'll see how much overlap there is in terms of small molecule approach versus large molecule approach. It should be a little bit more uniform in approach than discovery necessarily is, but that will be the topic for next week.
So that finishes our main topic. Can't mention it enough that this is week 10 of our podcast. Can you believe, John, we've been sitting here doing this for 10 weeks now?
Yeah, 3 more weeks and we'll be 13 and we'll be popping out acne all over ourselves.
Well, this week we got a real special treat coming up for our feedback and questions. Instead of having to hear John and I all podcast, we're going to introduce you to our marketing team. And as a very special event, Jeremy and Aaron will be doing the feedback and questions section. So, Jeremy and Aaron, you guys want to say hello?
Yeah, that's right. So this is Jeremy. I am one of those Midwesterners that they're always talking about at the beginning, I guess. And Aaron, would you like to introduce yourself?
Yeah, I'm Aaron. I live in Kansas City also, so in the Midwest as well, and probably the segment that is loosest on the mask wearing, because I never see anyone wear masks.
Really, Aaron? So and you live in what part of Kansas City? Are you in West?
No, I'm in Mission, Kansas, so about 10 minutes away from Westport. And of course, I'm pregnant, so we're very strict on wearing masks. And whenever my husband goes into a store anywhere, he wears them, and he always feels like he's getting like death glares. I'm like, who are you, and why are you wearing a mask? So
Okay.
I know.
Oh, it's not that bad out in the burbs. I know that we're not that bad here in Overland Park yet. There's no death glares. I think we're still not there yet. And by the way, Jeremy, you're soft right now for me. You're hard to hear, just as an FYI.
Oh, okay. Are you able to hear me right now?
Better.
Good. Okay, excellent. It's actually weird to have the role reversal here, guys, with you telling me how my audio is.
Yeah.
I'm over in Missouri, so as of I guess when this comes out, it will have been last Tuesday. The governor here opened up the whole state as though COVID never happened. So I'm experiencing COVID a little bit differently, certainly than John, but even more than Dom and Aaron right now too. So
you're saying it's like you walk into a restaurant, there's no roped off tables or anything. There's no spots for you to stand. Like even the restaurants here, you walk in, there's like a segment around a bar for you to stand in this 6 feet. None of that's happening, Jeremy.
Yeah, I mean they put up the pretense for a little while, and certainly different areas like St. Louis was hit pretty hard, and so I have been there, and they're pretty strict with it. But like I just took a little road trip around the state to meet with different associates. Just got back yesterday, and I can tell you in the majority of Missouri, certainly in southern Missouri. It's like COVID never happened.
And you know, Jeremy, that's close to Arkansas. And Arkansas just hit the critical state. They're now on the do not travel to state list as of today. That's breaking news. Arkansas is now back into like I don't know if it's lockdown's the right word, but they've now been put on the no travel to state according to the guidances that are out there.
Yeah, I mean, I had to take a photo of it to show my wife so that she would believe me. But I went to one small town shop on my way back up home yesterday, and there was a sign outside that said, "Please, no masks." And I thought, what?
Oh no.
Wow.
I could just feel John cringing through the like John's about to lose his mind. I could feel it, John.
Yes, so we need to send them a list of references to read and say, this is really what you need to be paying attention to.
Yeah, I'll send you guys the photo. I couldn't believe. I mean, it was an ammo shop, so maybe it was for security reasons. But I just thought I got a kick out of it.
Yes. Yeah, I saw that. I saw that. It was a baseball player this week who did like a YouTube thing from his car talking about how he wasn't impressed with the sheep who wore masks and all this sort of stuff. And it's like, look, just read a little bit about why we're doing masks and just check on transmission with and without masks, and just stop accusing people of being sheep because they're trying. They've done a little bit more research.
Yeah, I'm so happy that I've been able to listen to the podcast and you guys talk about all of the studies that you're reading, because I do feel like I have a little bit of ammo when people question why I wear a mask. I just pull out a couple of percentages and where I heard it from and feel a little bit validated.
Well, speaking, the latest is if you're sick, wearing a mask is a must. That is undoubtedly, there's no refuting that. If you happen to be infected or you're not feeling well, a mask is. So the mask, see, people have this misnomer. The mask is not to protect you, it's to protect the people around you. That's what people don't understand.
That's exactly it. And it, yeah, you're absolutely right. And actually, it doesn't need everyone to be wearing a mask, but it definitely needs a substantial proportion of the population to be wearing them, and that cuts transmission down. And it's it's out there, and to sort of denigrate people for wearing something that really you're benefiting from them doing it is not right. There's my little soapbox moment.
I like it. So, Jeremy and I, we don't have feedback or questions from the audience this week, but we thought it would be fun to surprise you guys with some questions that Jeremy and I have. So I'm going to ask the first one of, just because we're both curious about this, what were your first thoughts when we approached you about the idea of a podcast?
John, you want to go first or you want me to jump in on this?
Well, as a fully paid up member of the introvert family, I was a little bit horrified to begin with, but ultimately I'm always willing to pitch in if I can help, help the business. So it's one of those things, you suck it up and do it. It's not as bad as my absolute horror is actually doing presentations to any sort of size of audience. So this isn't anywhere near as bad as that. So once we got past the first one, I've been fine. So my first reaction wasn't great, but it was only a couple of minutes. I'm like, yeah, I can do that.
The funny thing is we're so different, John, because the first thing I thought was, what took you so long to ask me to do one? No, all kidding aside, I was excited, honored, thrilled, and even like something that I feel like we had been, Jeremy and Aaron, we had talked about it years ago when we were when you first came on, Jeremy specifically a couple years ago, like, hey, we could do these things, and that had always been mentioned, but. Yeah, no, I think it's funny that John had his and I was like, yeah, and the reality is, John, you're quite good at this, and sometimes I struggle, right? So
But I have this. So I think part of my issue, and this is why I don't like giving talks, is this constant. You get this constant feedback going on in your head, and I really hate saying something that isn't accurate, and it kills me when I do it, And every so often, I still give talks. I hate doing it, but I still do it. And then you remember that one thing you say, and I play it over and over and over in my head, and I beat myself up about it. So that was like part of my nervousness about coming into this. It's like, because I want to make sure I've got all my facts absolutely straight, and I don't want to say anything that anyone could ever disagree with. And it, yeah, I need to get over that.
well, what a treat this is. Keep them coming, guys. Wow, this is exciting. Cool little surprise.
Yeah, so the next one, I'll go ahead and take one here, Aaron. So we, of course, started the podcast because of COVID, and we've seen so much happen in the world of COVID just since even we started the podcast. Both of you have kind of anecdotally given references to experiences you've had in your various locations. I was curious, though, just a completely non-scientific question. What is the craziest thing you've seen in your personal experience related to COVID, whether it be somebody that just was flagrantly ignoring social distancing or anything like that at all?
So I'll go first. There are two things pop into mind. When this first happened, somebody walked into the grocery store with what looked like a World War II gas mask, or like it was an absolutely military grade gas mask, and that caught me. I'm like, man, there's no way we can walk around with these things on, and that's still a visual I have in my head. The two big filters on the front of it, and then the part that got me was just the smile on this individual's face that was like, I'm an engineer. I think he looked like an engineer of some sort. You get those feels. He probably had it from something, and he was wearing it like, look at all you fools out there are going to die, and I'm safe inside my mask. And I kind of had one of those moments of like, not I had a mask on, but I thought, man, that is complete overkill. And then just leading on that, the other was, I think I mentioned it when I was at Walgreens, and this dude just walked up right next to me while we were checking out, right? This was probably week 4 into things, and everybody's kind of like, keep your distance. And he just got right in. And that's when I was like, I think I looked at my wife and said something like, he's invading my space, and I really kind of, that one still stuck out where like, and even now when I'm out, that moment of when somebody just does stuff and you're like, are you just absolutely immune to the fact that there is still a pandemic going on? So those are my two stories.
So this is something that I didn't personally see, but heard it from my son, who heard it from a friend. To be honest, people around here have been pretty well behaved with all this, so there hasn't been much unusual to observe. But from what I hear, the local supermarket, there was a guy walking around with a woman's panties as his mask.
I've seen a couple of pictures on the internet of people wearing that.
I think people were a little bit taken back by that. But I think for me, the most amazing thing was actually right back to the early days when I had come to Kansas City just to catch up on stuff, and then it was actually the timing of us closing the facility to people who didn't need to be on site, and so I left the same day as everyone who was going to work from home went to work from home. But I flew from Kansas City to Philadelphia, and the plane was, I think there was 14 people on a plane that was normally however many hundred. But walking through Philadelphia airport was fascinating. I had enough of a time that I could walk from Terminal B, where I came into, out to Terminal F, but actually I went and explored Terminal A as well, which is the international terminal. And it's like you've got this enormous airport as a complete ghost town. And so virtually most shops were shut. a lot of the restaurants were shut as well, and certainly internally, I pretty much had the whole terminal to myself. It's just really, really interesting experience as part of all this. But yeah, I haven't seen too much unusual beyond that.
Well, I think it triggered one thought in my head, not the airport one, but the pantyhose. There was also a video of like a guy like sneezing on his hand and wiping it on somebody. That is absolutely one of the worst visuals I've seen yet. Is some of those stories that popped up on the internet.
Woof.
Yeah, and like he got thrown. I think it was because he was getting thrown out because he didn't have a mask. So like he like either sneezed or coughed into his hand and then wiped it on. Like this was at like a Walmart type store, a greeter, and I'm just like, whoa, people are just nasty.
Yeah, people are. I mean, there's actually a really tragic case in London where a woman who worked for London Transport, someone spat on her, and she basically developed COVID symptoms and died as a result of that.
Oh boy.
Oh, that's terrible.
Yeah, absolutely. Yeah. Any more questions?
Do you want to take the next question then?
So I do have one more question because I know you guys both love this topic. Dom can't say pizza. That's the only thing. But what are your favorite foods to cook at home besides pizza? Because we do know Dom loves his pizza.
I can go first here. My if it's not pizza, I make a wonderful baked stuffed lobster.
Ooh.
And I use scallops and a mixture of Velveeta cheese, actually, and some Ritz crackers. And I make a delicious baked stuffed lobster.
Yum, Velveeta cheese.
And it's lobster. It's not a lobster. It's a lobster.
Yeah, my cooking probably isn't what it would be if I had a better supply of fish. It would be completely different. One of the dishes that has stuck with me since childhood is actually soused mackerel, which is soused mackerel is basically this is a dish that basically harks back to my childhood. It's not. I don't know. It's necessarily my favorite thing to cook, but it is a real highlight whenever I do it. And basically, it's mackerel. It can either be done cold, but you're using vinegar to cook it, and you also pickling spice and onions in there as well. You actually serve it cold, but it has once you finish cooking, it has the most wonderful smell. I remember as a kid going to the fridge when my mum made it, and you'd open up the pot, and there's just this wonderful waft of pickling spice and vinegar, etc. So I do that a lot. But actually, probably our most successful thing that we routinely do when it comes to Christmas Day. We don't do like a full Christmas meal anymore because there's just too much going on. We're too happy drinking wine and stuff. But we actually have gone the way of doing a full English breakfast for the kids, the whole family, and it's become a big ritual now. That's basically eggs, bacon, sausage, tomatoes, mushrooms, baked beans, toast. You obviously have a pot of tea that you're drinking with it, and that dominates our Christmas Day, and that's probably the best thing we are at doing.
Yeah, that sounds delicious.
Aaron, maybe I can help out. How about chicken parmesan? You want me to send you that? You think you can do that one?
Oh, yes. That's something more along our lines. That sounds delicious.
All right. Maybe I'll even donate. Too bad I can't get to you fast enough. I'll give you a jar of pasta sauce too. But no, chicken palm's another go-to.
Even better.
Great question.
All right, so last one, guys. Real quick, real simple. Favorite podcast episode so far?
Oh, all of them. No. Let's see. I feel like it was for me. It was, I think, number 3 when it was on antibody testing, and that's probably the one that sticks out for me.
I actually really enjoyed the one where we basically said nothing because Don and Frank did it all. It was a good chance for us to sit back and listen, and it was I like the thought of having more people in to interact with like that, because I like we're doing here. It's really nice to, much as I enjoy talking to Dominic, it's really nice to also interact with other people on this.
I guess I should have caveated. It's a given that one is probably our best, but I was trying to be, and no, like that. I do think that particular one stuck out because when I was listening to it, there were moments where I was like, oh wow, this person sounds like they really know what they're talking about. Like there was some passion involved when vaccines and antibodies, right?
Well, it has been a pleasure listening to you guys, not just producing these, but also Aaron and I get a kick out of editing them, mixing them. We go back. We've both learned so much, and I know that there are listeners out there. We've now passed the 2,500 views, listens mark, and to just watch it grow, and obviously it would not exist without you guys. So from Aaron and I both, thank you so much. It has been a pleasure to even be part of this so far, and we're looking forward to many, many more episodes.
Agreed. It's been very fun.
And well, thank you. And that does it for this week's podcast. For next week, we're going to continue in our series of where does bioanalytics fit into the drug development process. Next week's episode will focus on the clinical side. So that'll be part 2 of 4. Any last words, John?
I'm very much looking forward to talking about the clinical piece, because I think there's tons we can talk about, and it'll be good and entertaining, I hope.
That sounds great. And then I hear you got tomorrow off, right? You got a long weekend?
So our excitement this weekend is we actually have a dumpster in the driveway at the moment. So we're going to empty our garage, which has got a ton of stuff in there, hopefully to free up some space so we can properly store things. And at some point, I'll talk more about our garage, because it's actually storing. At the moment, it's storing a 1971 car, which shall remain nameless, that I hope to get restored soon. But that will be part of the long-term tidy up of the garage. Get that out for restoration, then I'll talk more about it.
You're paying, it's always funny when you take time off to work. Well, that'll do it. Thank you, everybody, and have a wonderful week.